Therefore, consumption of blueberry has been recommended as part of daily healthy eating style. So this is really a wonderful news for me to step into this breeding position, and it's good news for the industry because our consumers, even including us, we understand how beneficial blueberry is, and with the market or the consumer demands increase, which is still increasing, and we all can do as a team a better job to produce better blueberries and more blueberries to meet the need from our consumers. So blueberry has a huge impact for Georgia economy, and we are ranked top 10 Georgia commodities, and blueberry has been shown to have an impact of $300 million in year 2021. Blueberry is in the family of Ericaceae and in the genus of Vaccinium, section Cyanococcus. And in this particular section, there are 9 diploids and 12 tetraploids and 3 hexaploid species. So for the blueberries that actually grown commercially, they largely fall into 2 categories or 2 species. One is a tetraploid species that including commonly grown here, southern highbush blueberry, which has like medium chilling hour requirement from 100 to 600 hours. And the southern highbush has tremendous benefit and has large acreage right now because it's ripe. These kind of species ripes earlier and have very smooth texture, large berries and very thin skin. It just have good, very good fruit quality. And this tetraploid species also including northern highbush, which have a bit higher chilling hour requirement from 600 to 1200 chilling hour requirement. And also including low bush, which mainly produce in the colder region like in Canada and Europe. Now, commercially grown blueberry also include hexaploid, which is known as rabbit-eye blueberry. Rabbit-eye blueberry is native to Georgia. And actually, the breeding program cultivates that blueberry from the natural selections. And therefore, because they're native, they also have extreme vigor and longevity of the orchard. So one thing about southern highbush grown in Georgia is that their orchard life will be limited. You know, if you manage really well, you probably can do 15 years, but sometimes you just can't help, and they have to have a turnover maybe between like 10 years, 9-10 years, whereas rabbit eye can last many years and still producing. Because rabbit eye also have strong tolerance to heat and requires much less of the soil amendment. So as a new breeder, my vision for the blueberry breeding program and UGA is to support the overall, our overarching goal is to support the sustainability and productivity of blueberry cultivation in Georgia. So specifically, because I am the 4th blueberry breeder, we have a lot of genetic materials in our program. So the first thing I would like to do is to continue the selection and advancement of current elite breeding lines. But in the meantime, I would like to broaden the genetic base of blueberry breeding by recruiting additional locally adapted diploids and hexaploid species. And I would like to introgress, like I said, to improve southern highbush or improve the fruit quality of rabbit eye, to introgress vigor and local adaptability from rabbit eye to southern highbush, and through interspecific hybridization. I would like to produce a diverse set of elite blueberry cultivars adapted to local growing conditions. And these blueberries also could be packaged with desirable genetic traits such as fruit size, color, flavor, quality, disease resistance, and machine harvestability. And I also have an intention to work with scientists in the other regions of the United States to engage in state-of-the-art genomics and genetic tools to develop molecular markers and accelerate breeding cycle. So I'm going to share with you some of our ongoing research and breeding activities for the past year. So the first thing I did, as I said, this is a legacy program. So the first thing I did was to consolidate the pedigree of our UGA breeding history dated back to 1950s. And we have entries of over 3,000 right now, 4,000 selections in the UGA breeding program. And however, our program in history does not do any interspecific hybridization. It's all just, you know, highbush by tetraploid within the tetraploid level or within the hexaploid level. The distribution of our program by species is like we have rabbit eye right now is about 30%, and southern high bush we have about 61% in our selection. And this is really not by species, but we also have a special category. It's accounting for 8% of our selection. They're called ornamentals. So these are edible ornamentals. They do not grow like as a commercial blueberry, so they are small in stature, or they have the characteristics that actually for home gardening or for landscape. So we have a small portion of germplasm in that category. So as my predecessor, immediate predecessor, Dr. Scott Neesmith is still with the program, and so he provided me the seeds when I first started back in 2022, and then we were able to germinate the hybrid seeds from him. So you can see here are the germinated seeds these are 72 cell trays, and then we moved them to 15 cell trays to boost up the growth, and then we have now from 17 crossing family, we have like about over 2,000 seedlings. We are planning to put them into the Lapa Research Farm somewhere between March and April this year. So this will provide new seedlings for us to continue more genetic material and for selection. In the meantime, I was trying to learn how to do crossing, and it's really fascinating. So I was really delve into this learning experience, and I practiced over 5,000 pollinations, and end up to did about 65 different crossing combinations. It's just fascinating. I came from the peanut breeding background. It's breeding just part of my job description anyway. So blueberry is actually easier than peanut. So the word of peanut, when we do crossing, when we do one pollination, you know how many seeds we can get? I don't know if anybody know peanut, but we just usually get one seed. For those people with like a green thumb, whatever you call it, they may get 2 or 3. But then with a blueberry, it's just so much easier. With one crossing, you get from 10 seeds up to 80 seeds. So this is really much easier, and it's so much fun. So we're able to make over 5,000 pollination from 65 different combinations, and these are including intraspecific and also interspecific with trying to introgress traits for southern high bush and rabbit eye. So one cross that we made is between rabbit eye and southern high bush. So the line, the TH, so the line TH2944 is a southern high bush, and the Kluwer is a rabbit eye. So we made a cross, and then we also get a flow cytometer, and we were able to get a protocol right, and then we were able to compare to the rebel, which is southern high bush, and this is the F1 progeny from this cross. It is a pentaploid. So pentaploid, this is how they look like. It's really cute and very strong, robust. So we expect pentaploids to be largely sterile. However, at this higher level of ploidy, blueberry is quite forgiving. So they will produce some probably viable pollen, which will help us to bridge the gap between the ploidy level of southern high bush rabbit eye. So we will be able to, not for commercial, but for breeding purpose, we should be able to introgress through these vigorous seedlings. But of course, we have to select the ones with some pollen viability. So we also engage in blueberry tissue culture. Our target is to do some micropropagation of rabbit eye selection. Our growers made some selection, so we put them in tissue culture. They're actually, so this one is a rabbit eye, and this one is a Phascatum diploid species. Actually, we are trying to introgress traits with, so we're culturing, we were able to culture both. And for the Phascatum, we intended to double the chromosome of this and to make it more cross-compatible with southern high bush. And this work is done by my master student, Emily Walter. That's her master research project. For the chromosome ploidy induction, we're using colchicine in vitro treatment. So this one, it shows you we are treating emerald and rebel because Dr. Oliver gave us some of these southern high bush already in tissue culture. We just give them a try. So after culture treatment, we can see that the new growth, this is not very clear, but these are pretty much killed. I mean, the colchicine is toxic, but these new growths have high potential of ploidy induction already because colchicine is a mitotic arresting agent. So this is our nursery that we, so the history is that Dr. Neesmith is based in Greenfield campus. So he, for his past about 30 years of research and breeding, his breeding effort for Blueberry, most of his stuff is in Greenfield campus, which is about 2 hours north of Tifton campus, where I'm located. So when he retired and when he was consulted for establish this continuation of his research for this position, he insisted to have this position placed in Tifton campus because our research from Alapaha is only 30 minutes away from Tifton, whereas for Greenfield, they have to drive 2.5 hours. So with that, so most of the genetic material and facility are in Greenfield campus for breeding purpose. Oh, whereas I just have strong voice, I guess. Whereas Tifton have nothing because that's his research effort in Greenfield and Alapaha. So when he moved this program, so we have nothing. We have no nursery, no greenhouse, no nothing. But our administrative team are very supportive of us, so gave us a nursery. So now we were able, that's why I'm showing you this nursery. It's just from scratch, from ground zero. Dr. Nismus would transport his material to Greenfield, so we could have material with the breeding and other efforts. So we literally received over 1,000 potted plants from a Greenfield campus to put in this nursery. Oh, the picture here is my technician. She was hired about 1 month later than I started, and she was very instrumental getting all these things going for the breeding program in Tifton. And we also have this shade house. We had this shade house to do the cuttings because we have new selections, and this is temporary because we just started. And just 3 days ago, we encountered a severe weather in Tifton, and the wind was very strong. So this structure is already becoming history. It's being lifted up and piled on top of our plants. But the good thing is, we already have another shade house that is more permanent, and that one stands through the weather, so which is good. Yes, we have a much better and much larger shade house area. So this structure is conducive for us to do cuttings and to continue propagate the selections. So Alapaha Farm in UGA was acquired back in 1950s when Dr. Brightwell initially started, and that farm has over 50 acres of planting. So right now we have 17 acres of planting over close to 1,500 different selections. And we did phenological study of all those lines, and then among the 500 selections, we can see a wide distribution of 50% blooming time and fruiting period, which serve as a good genetic resource for us to continue the research on the genetic control of these phenological traits. We also work with Dr. Josh Clevenger at Hudson Alpha Institute of Biotechnology, and they did whole genome sequencing with the scheme sequencing for us of 51 blueberry genotypes. The average depth of coverage is 3x, and we were able to recover 1.5 million SNPs, and we used a subset of 70,000 to do a phylogenetic analysis. So here shows the phylogenetic trait. So all these blue circles are southern highbush, actually circled by crossing families. So this is correctly clustered, and this green circle is rabbit eye family. So these markers with such a shallow depth were able to properly classify the species and also the families according to breeding history. It's pretty amazing. So as a first year, as I said, first year look at the blueberries, and I think I'm very dumb. The first thing I look at, oh, the berries are so big, yeah. So I took a picture of the big ones. So this one line had jumbo fruits, and you can see this is actually on farm, which is using that meter. Everybody knows to look at the size. This is ranging from 18 to 22, and this is a jumbo a rabbit eye, 26 millimeter. And this is so our growers selected some of the lines late in the season. So this is a high yielding breeding line. So you can see a lot of fruits already on the ground, but it's really high yielding and it tastes good, also large in size. Lastly, I would like to introduce to y'all our college decided to set up a Nismus professorship for blueberry breeding. So as I said, Dr. Nismus retired in 2019. And he had 30 years of distinguished career in blueberry breeding. And this endowed professorship will provide funding in perpetuity of blueberry breeding and cultural development, and also sponsor student training. So I'm hoping that y'all will be interested in sponsor this professorship that will make us to be stronger and have a longer term investment, like in every way, to make better blueberries for Georgia. Thank you. I don't know if I have time to take any question.
Thank you so much, Dr. Juliet Chu. Our next speaker is Dr. Rachel Mallinger from the University of Florida.
Thank you. Sure, yeah. All right, I will try not to wander too far from the microphone. If I forget, just yell at me. So I'm going to be talking today about pollination. I was here last year with you all and spoke primarily on the pollination requirements of blueberries and different cultivars. Today I'll be taking a little bit of a different approach and talking to you about different insects, bees, that pollinate, their role, some of the data we have on their unique roles and how they work together. And I'll end with some new research that we have on flower traits, looking at flower morphology and nectar production across different cultivars and how that might affect pollination. And I'm at the University of Florida. All right, what was that? Go Gators. Go Gators, that's right. Okay, so I'm going to start with just a brief background on pollination. For some of you, this will be a review. For some of you, this may be new. But pollination is essential for yields. Pollination is the process by which flowers become fruits. And the pollen is produced by the anthers, which are those brown, orangish-brown looking structures inside the flower there. And that pollen needs to move to the tip of the stigma, which is the tip of that green structure you see inside the flower. And you can see in the picture, let's see if I get, yeah, you can see that there's kind of a glistening, sticky material at the tip of the stigma. That's where the pollen needs to adhere. And as you might imagine when you look at this flower, that movement from the anthers here to the tip of the stigma is not very effectively done by things like wind, because the flower is enclosed in the corolla, the petal structure. And so it's typically done through insects. There may be some amount of pollination that happens just by gravity and agitation, but again, wind is very ineffective. So even for pollination that happens within the flower, pollen moving from here to here, even just that short distance really relies on insects, and that's a point I'm going to hit home again. It's not just moving pollen from flower to flower, but even within that flower, the pollen is not readily going to move from here to here. Again, that's in part because those structures are enclosed in the petal tube, so wind is not effective. It's also because these anthers here don't readily release pollen. They require a buzz, a particular vibration of a particular frequency in order to release pollen. And bees provide that buzz to release the pollen. So these pictures here are from a colleague in Michigan. And this shows what your fruit set would look like without insects. This was done by bagging the flowers to avoid insects. And then what your fruit set would look like with insects. So again, some amount of pollination might happen without insects, but you're going to have very small fruit. And again, that pollination can happen within a flower, just moving from anther to stigma, or between flowers on different plants. So when we refer to self-pollination in blueberries, we're referring to pollination that happens within an individual flower. So going back, a flower like this. But self-pollination can also refer to that between flowers on the same plant. So pollination that would happen from here to here would also be self-pollination. Cross-pollination is pollination that happens between different plants, and ideally plants that are not genetically identical, so different cultivars. So again, pollinators are going to assist with that self-pollination, even within an individual flower, as well as cross-pollination. Okay, so I'm going to move on to talk a little bit about our research on the roles of different pollinators. We have 4 main insect pollinators in the southeast. The main pollinators are bees, because again, these can provide that buzz of that particular vibrational frequency to release the pollen. You might see other insects on flowers, butterflies, wasps, and those might do a little bit of pollination, but they're less effective. So our main pollinators include bumblebees, carpenter bees, southeastern blueberry bees, and honeybees. You might think that these 3 pictures are the same pollinator, but they actually are 3 different pollinators. And so we have bumblebees on this far end, carpenter bees, southeastern blueberry bees, and honeybees. All right, so carpenter bees, what do we know about carpenter bees? I don't have any data from my lab on carpenter bees, but there has been some data from other labs that I'll share with you. So carpenter bees nest in wood. They like to excavate nests in wood. So they're one of the few bees that can be a pest problem, particularly in Georgia and north in the southeast. They are generalists, so they'll visit blueberries, but they'll visit a variety of other types of plants as well. They have long foraging periods. You'll see them active throughout spring, summer, and fall. And they are what we call primary nectar robbers in blueberry. And so what they do is they create these little slits or holes to access nectar. So they'll put their mouth part through that slit or hole that they create to get the nectar. They do not typically forage for pollen in blueberries. They typically just want the nectar. So that's why they do this. It provides them easy access to nectar without getting pollen on their bodies and without having to do the work of coming in from the top of the flower. This also creates a slit or a hole that then honeybees can use. Honeybees are known as secondary robbers. I'll mention that again later. But the honeybees go in and access that hole to nectar rob. But the good news is that studies from a while back in other parts of the southeast have found that carpenter bees can contribute to pollination. Even this nectar robbing does apply some agitation to the flower to release pollen. The bee gets some pollen on its mouth part when it sticks its mouth part through that slit, and so some pollination does happen. It's less effective than what we call a legitimate visit, which is coming in from the tip of the flower, but it does contribute to pollination. The other thing that this research found is that nectar robbing can make the whole field a little more attractive to honeybees because they like to then use these slits or holes, and so it can actually increase honeybee visitation rates. So they are neutral to beneficial. This is not necessarily negative for the crop. However, this research is pretty old. You can see it was from 2004, and there have not been too many studies since then to look at in our newer cultivars how this nectar robbing might be beneficial or detrimental. So then the other wild pollinators we have are the southeastern blueberry bees. These are specialists of blueberry and related plants, so they forage primarily on blueberry and its relatives. And because of that, they have a short adult foraging period that coincides with blueberry bloom. You'll only see them in the spring. They're solitary. They don't have large colonies. Each female has her own offspring, and they nest below ground. So because of these attributes here, there's been limited work on domesticating them. It's hard to domesticate a solitary ground nesting bee, which is why you can't just purchase them and release them to your farm. Unlike carpenter bees, they collect both nectar and pollen, and they don't do any nectar robbing. So what we found, I'm going to show you a few graphs for some of these pollinators here, and what you're looking at, each dot is a row in a farm, and we did this work over 21 farms. So you can see 21 times however many rows we looked at per farm. That's what each dot is. We have the number of visits per row that we recorded just walking down the row at a quick pace. We have proportion fruit set, which is estimated based on the number of flowers we counted and the number of fruits we harvested, and then the yield per 100 flowers down here. And what we found is that as the visitation rates of the southeastern blueberry increased, we did see increased fruit set and yield. And this comes out to about a 6.1% increase in percent fruit set with each additional visit, and an increase in yield of 10.51 grams per 100 flowers. You might note that visitation rates are relatively low, but again, this was done by walking at a quick pace down the row. Many farms do have zero, so you'll note there are a lot of dots here on the zero, but we saw higher fruit set and yields on average when we had more southeastern blueberry bees. So what are they limited by? We looked at a bunch of farm characteristics across these 21 farms, and we found that farm size, as well as the landscape surrounding the farm, did not seem to influence how many southeastern blueberry bees we found. The one factor that did pop out was management. So we had over twice as many southeastern blueberry bees on organic farms compared to conventional. I think this makes sense. These bees are specialists, so they're not benefiting from other habitat in the landscape, and they're pretty much spending their whole life on the blueberry farm. So the management, including insecticide use on the farm, is likely going to be the biggest factor affecting their populations. They're not going to benefit from diverse pollinator plantings or necessarily natural habitat off the farm. And they're going to be highly sensitive to pesticide applications because they forage only on blueberries. All right, so bumblebees are another pollinator that we have in both wild and then also managed form. So there are managed bumblebee colonies that you can purchase that I'll talk a little bit about, but we also have wild bumblebees. Like the southeastern blueberry bee, these bees forage for both pollen and nectar. Unlike the southeastern blueberry bee, they are generalists with a long foraging period. So they're active throughout nearly the entire year. They forage on a variety of plants, but they do seem to show a strong preference for blueberries. They are social and they nest in cavities, which makes them easy to domesticate, and hence we have these managed colonies that you can purchase. So what we found is that these are effective, slightly less so than the southeastern blueberry bee. So here we have, again, bumblebee visits per row per period. You see higher numbers, so we have more of these compared to the southeastern blueberry bee. We also have a positive relationship with our estimated proportion fruit set and yield. Again, these numbers are a little lower than they are with the southeastern blueberry bee. So each additional visit percent fruit set increased by 4.2%, but there was a very small positive relationship with yield. So what did we find in terms of what limits their populations? There were two main factors that limited the number of bumblebees on farm. First of all, what we found, and that first point here is not shown in the graph, but what we found is that bumblebee visits did significantly increase with the amount of natural habitat surrounding the farm. So of our 21 farms, farms that had more wooded habitat or open grassland habitat had more bumblebees. Again, I think this makes sense. Bumblebees forage throughout the year, and they forage on a variety of plants. So they're going to benefit from other beneficial habitat in the landscape. The other limiting factor is stocking density of the managed colonies. So we found that bumblebee visits did significantly increase with colony stocking density. In Florida, growers typically use anywhere from 0 to 1 quad. Each quad has 4 colonies. So here we have up to a single quad, but that's 4 colonies. And as stocking density increased, so did the number of visits in the crop rows. All right, and then finally we have honeybees. So these are non-native, so they only exist in managed form. In some places, like in Florida, we do have a lot of feral colonies as well. Like bumblebees, these are generalists with long foraging periods. They'll forage on a variety of plants and throughout the year. I mentioned before that they use those slits made by carpenter bees. They're secondary nectar robbers. They actually don't make the slits themselves. They just utilize the slits that other bees make. They primarily forage for blueberry nectar, but they do sometimes forage for pollen. So what we found is that these are effective as well, sort of on par with bumblebees. They did not have quite as strong of an effect on percent fruit set, only 0.6% increase per visit, but they did have a stronger positive effect on yield compared to bumblebees with 0.73 grams per 100 flower increase per visit. So in terms of ranking, southeastern blueberry bees were the most effective, and then honeybees and bumblebees followed. Again, you can see that we had much higher visits per row compared to our other pollinators, and there's that positive relationship with fruit set and yield. In terms of what limits honeybees on farm, they're not really limited by characteristics like landscape or by farm management necessarily, because farmers bring them in. They exist in managed colony form for the most part. They don't have wild populations. So they're primarily limited by the number of bees that are brought into the farm. But what we found is that there was not a strong relationship with honeybee density in terms of colonies per acre. Growers in Florida using anywhere from 1 to 12 hives per acre. So there was no real relationship there. But what there was a relationship with is the hive size, the number of bees inside the hive. And so it's not just about bringing in more hives, but bringing in hives that have more bees. And I'll show you next a little bit how you can assess the size of the hives that you're bringing in. So again, no relationship with stocking density, because there was a lot of variability in terms of the size of the individual hives. We actually found many hives that didn't have any bees at all. We call these deadouts. So there were a lot of empty hives on the farms. We also think that how many honeybee visits you get might be limited by their attraction to the crop. but that work is still ongoing. So again, the primary limiting factor is the size of the individual colonies, not so much stocking density. So how can you assess hive size, given we found that this is very important? We've been looking at two different ways. One is to count returning foragers in a 1-minute period, doing this during bloom, ideally peak bloom, and on a good weather day. The entrance to the hive is typically down here at the bottom of the hive, but there can sometimes be other entrances as well. So you watch this bottom slit down here, and you count how many foragers return in a 1-minute period. We found that average to above average counts were around 50 bees per 1 minute, so close to 1 bee per second. If you're seeing 20 bees per minute or below, that's a smaller than average colony. But that again is quite sensitive to weather as well as foraging behaviors, but it's relatively easy for a grower to do. You can wear a bee hat or a bee suit if you want. You can stay at a distance. The best way, however, is to do what we call a cluster count. Typically, you'd want to do this with a beekeeper and with beekeeper permission, but you can look at the hive from the top or from the bottom. You don't have to pull out the individual frames. You're just looking at the top or the bottom. From below is less sensitive to weather, so that's what we recommend. So that would be lifting the hive box up and looking at it from below. What you're counting are the number of interframes covered with bees to the nearest half. So an interframe is like this here, this kind of open space in between a frame, and you're looking at how much of that interframe is covered in bees. The margins count as a half even if they're full. So this here would count as a half if it's full, and this here would count as a half if it's full. So what does that look like? Here, this is 7 interframes. So we have 1, 2, 3, 4, 5, 6 that are full, and then 1 that is partially full, and then a little bit here, so we count that as 1 combined. Over here, however, we see far fewer bees. So we have 4 interframes covered in bees, but each is only about half covered. So 4 but half, so we would count that as 2. So this is what 7 would look like, this is what 2 would look like. Here is a strong or large colony that has 9 interframes covered with bees. Again, the margins count as a half, so that would be 1 total, and then 8 in the middle. Some work that a former postdoc in my lab did found that the number of interframes covered with bees is significantly highly correlated with the actual number of adult bees. So he did this by actually extracting all adult bees from the hive and then comparing that to the number of interframes. So you can see anywhere from 10 to 50,000 bees, and that's significantly positively correlated with the interframes, which can vary from 0 to 10. We saw examples of a 2, a 7, and a 9 in those previous pictures. You can also do quality control for the bumblebees that you might bring in, but it's important to note that bumblebee colonies have around 200 workers compared to 10,000 plus. Going back to this graph, you can see 10,000 is actually on the lower end. And so when you're doing 1-minute counts, you might only find 1 or 2 bumblebees returning compared to 50 or 60 in a honeybee colony. Bumblebees also spend a significant time in the field, which means they return less frequently. So you can do the 1-minute counts. Typically, I just recommend to growers, if you see something in a minute, like even a single bee, that's a good sign. You can also close these plastic doors here and lift the cardboard box and see if there's some activity in each of the 4 colonies in the quad. Again, you'd want to close those plastic doors first so you don't get stung, and then you can lift this box. So again, I typically just recommend ensure that they're not dead. Any activity at all is a good sign. Keep in mind that these are much smaller, so you're not going to see the number of bees that you do in honeybees. All right, so I'm going to move on to talk a little bit about what we found in terms of the different roles of these different bees. So this work, again, was done across 21 farms. Each dot is a crop row, and we did this throughout the bloom period. So what we found is that honeybee visits to the crop field actually didn't vary much throughout the season, from early to late, pretty consistent. But we found significantly more bumblebees and highly significantly more southeastern blueberry bees in late season. So phenology is a limiting factor, especially for the southeastern blueberry bee. They come out a little bit later in the spring. Many of our cultivars are bred to bloom very early, so there's a little bit of a mismatch there. For the bumblebees, this includes both wild and managed. So the managed bumblebees are probably making up a lot of these early visits, but it's not till later in the season that we start to see a lot of wild bumblebees. And so that's why you see that positive relationship there. So one of the ways that these bees can complement each other is by being active at different times during the bloom period, with managed honeybees making up almost all of the visits in the very early bloom, along with managed bumblebees. And then later in the season, you start seeing wild bumblebees and southeastern blueberry bees. Other work that my PhD student John Turnist has done looks at the different role of these different bees and what happens when you use them together. So to do this work, he caged blueberries with either just honeybees. So this is a cage of blueberries. You see the mesh on the outside. So these blueberries are just receiving honeybee pollination. And then he had other cages where he put in bumblebees. There's that quad box there, along with honeybees. The numbers here are total bees, so honeybees and bumblebees combined. 2 cages that only had honeybees and cages that had bumblebees plus honeybees. So you can see that visitation rates were overall higher when both bees are used together. I think there's a few explanations for that. I'll show you in a minute that the bees interact with each other, which might increase visitation rates. We also know that bumblebees have a higher affinity to blueberries, and so they forage a bit more readily on blueberries compared to honeybees. And we also found that under these cooler temperatures in particular, there was a bigger gap between using just honeybees and bumblebees. And that's because bumblebees will forage under these cooler temperatures, 60 to 65. So down here in the 50s, we see overall lower visits in both types of cages. And then up here, when we're at 75 plus, this gap is narrower. So we still see more visits when you're using bumblebees and honeybees, but the honeybees are approaching the combo. So there's less of a gap here at warmer temperatures. The biggest gap is in these cooler temperatures here, because we know that bumblebees are more able to forage under those cooler temperatures. So bumblebees increase floral visitation, overall floral visitation, particularly under cooler temperatures. As I mentioned, John Turnist, the PhD student who did this work, he also found that there were more interactions when we had bumblebees and honeybees together. So these are the bumblebee plus honeybee cages, compared to when you have only honeybees. And what I mean by an interaction is an actual sort of collision between bees. So we saw about 0.74 collisions per observation period when we use them together, and 0.22 when you only have honeybees. So why is this important? Why do you want bees colliding and interacting with each other? Other research has found that when that happens, bees actually become a little disoriented, as you might imagine, and they're more apt to forage in different rows. They are more apt to move locations and forage in other areas of the farm, and this could increase cross-pollination. So the more interaction you have between bees, the more they might be likely to cross-pollinate by moving to other cultivars or other areas of the field. All right, and I'm going to end by telling you just a little bit about some upcoming research that we have. Another thing we're going to be looking at is how different flower cultivars might be more or less attractive to different pollinators. So we're thinking that some pollinators, like honeybees, might prefer certain flower morphologies, whereas bumblebees might prefer other morphologies. And this might explain why we see in the field, we see some areas that have more honeybee visits, some areas that have more bumblebee visits, and some areas that have more visits by the wild southeastern blueberry bee. So the first step has been to look at the variation in flower morphology across cultivars. So each picture here is a different cultivar, and you can see that the morphology is quite variable. So some of the things that we're looking at include the length of the corolla, which is the length from here to here. We're also looking at the width, measured kind of at this midpoint, as well as the width of the opening. And it might be the width of the opening that is particularly important for determining what bees visit and how frequently those bees visit. You might also note that another interesting morphological difference is the length of the stigma here. So in some cases, it actually protrudes from the corolla, from the petal, and in other cases, it's completely hidden within. And so we're really interested in looking at how that affects pollinator attraction as well as pollination. I'm showing two kind of extremes here, a long, narrow corolla and a shorter, wider corolla, a stigma that is concealed within the petal tube, and one that's protruding. And then finally, we're looking at variation in both pollen and nectar production across these cultivars. So this is total nectar sugars across cultivars, measured at 2 different time points. This is just before the corolla fall, and this is right after the flower opens. So just before the petal falls, this is the total amount of nectar sugars that flower produced. And so you can see that the total amount of nectar sugars that the flowers produced over their lifespan, right before corolla fall, was highly variable across cultivars. We have northern cultivars here and southern cultivars here. And so this different nectar production might have consequences for which cultivars are attractive to pollinators. Keep in mind that some pollinators are foraging for both nectar and pollen, so they may be less sensitive to the amount of nectar produced, whereas other pollinators like honeybees that are foraging only for nectar might be more sensitive to this variation in nectar production. All right, I'm going to wrap up. I have a few more minutes. So I'll wrap up here and then take some questions. So in terms of working together, we found different bees are active at different times in the season. Bumblebees are more active under cooler weather. We think that the diversity of bees might increase cross-pollination because of those interactions between bees, forcing them to forage across different cultivars or areas of the field. And different bees might also visit flowers with different morphologies or nectar sugar amounts. And in conclusion, we've found across all of the results that I showed today that all bees contribute to pollination, but unequally, with southeastern blueberry bees being highly effective, followed by bumblebees and honeybees, both effective as well. And different bees are limited by different factors, so including management, the surrounding landscape, stocking density, and hive size. Different bees are active at different times of the year, under different weather and on different cultivars. And interactions among these bees might increase cross-pollination through greater movement across cultivars. And with that, I'll leave my email up. If you have any further questions, you can contact me, or I'm happy to take questions now if there's time. Yeah, Ryan.
Well, first I want to say, Dr. Mallory, you always do a great job. Learn something every time you present.
Oh, good.
Really appreciate you and your team.
Yeah.
I thought it really interesting what you're talking about with the frames in terms of high quality.
Mm-hmm.
That's really simplistic, and I like it.
Yep.
The problem is some of us have a lot of frames out
Yeah.
in the field, right?
Right.
And we only have one beekeeper.
I know, yeah.
It would take a long time to evaluate all the hives.
Yes.
Is there a way to try to correlate what you're seeing on top of the frames versus the visitation?
Mm-hmm.
Is there a 90% correlation? And if my guys sit outside a hive and,
Yep.
and time it for 5 days,
Yep.
and they do a bee count,
Yep.
can you get a correlation of what that means
Yes.
in terms of
Yep.
high quality?
Exactly, yeah. So Ryan's point is how correlated are like those 1-minute counts outside the hive to the interframe counts. And we have found a positive correlation. It's not perfect, but there is a positive correlation there. And so that tells us that those 1-minute counts are a pretty good indicator. It's not a perfect correlation because the 1-minute counts are a little more sensitive, not just to weather, but also to the colony foraging needs. Sometimes the colonies are just going to be foraging more than they are at other times. And so when you open the box, you're getting an idea of what's in there, right? When you're just counting the foragers, it might just be that they've got a lot of nectar stores, they're just not foraging for whatever reason, but there is a positive correlation. And so I think that doing the 1-minute counts is pretty good. That's what we recommend to farmers. It's not realistic to do the, to do the frame counts. You could, or the cluster counts, you could kind of split the difference and do the 1-minute counts across a larger number and then, maybe pick a subset to do the cluster counts. There's also some work. I was talking to another farmer that might be able to use thermal signals. So using like a sensor outside the colony that looks at the colony temperature as an indicator of how many bees are inside there. And so there's some work that's being done to see how well that's correlated and then what kind of a device you might need to get a high quality thermal signal. But that could be pretty promising too, because you could just, kind of go up and take essentially sort of measurement of the hive temperature from the outside. But I would say for now, yeah, we do know that the 1-minute counts are positively correlated, so it's a pretty good indicator. Yeah.
The quads are now being sold with an applicator of fungicides. So the quads are now being sold with an applicator of fungicides. Does that have any effect that you're aware of on pollination?
Yeah, so the fungicides that you're saying they're coming with the quads and to apply to the colonies themselves? Interesting. I do not know anything about how that might affect the activity of the bumblebees. We do know that some fungicides have what we call sublethal effects on bees. They don't kill the bees. They're not designed to kill insects, but they can affect things like their memory, their navigational abilities, their immune systems. So I would hope if the colony is recommending that you use that to control fungus, they've tested it and found that it has no negative effects on the bees. But that's interesting. We have not looked at that. But I guess it's a warning in terms of using fungicides in your field. They're probably not going to kill the bees, but just be aware that some research has found that fungicides do have negative effects on kind of the immune systems of bees and their behavior. Yeah.
Rachel, thank you for your talk here. On the organic side of it, I know you showed the blueberry bees having a higher percentage of being out.
Yep, exactly.
Is there any correlation to that as far as like honeybees in the field?
Right, with honeybees? Yeah.
Organic production versus like conventional production, just
Yes.
naturally.
Right, exactly, yeah. So we did not find that there was a direct correlation between the number of honeybees and the number of southeastern blueberry bees in the farm. And then in terms of how many honeybees were brought, there were slightly fewer honeybees on organic farms compared to conventional, but the difference was not terribly high. So I think it was on average 3.5 to 4 on organic farms, and more like 4.5 to 5 on conventional. So there are slightly fewer honeybees being brought to the organic farms, but again, it's not a large gap. And when we look at just the number of honeybees in a row and the number of southeastern blueberry bees, we did not find a significant correlation. So that tells us that probably the southeastern blueberry bees are not highly affected by the number of honeybees that are being brought to the farm. All farms are bringing a pretty high density, at least in Florida. We have anywhere, I showed you, anywhere from 1 to 12, but a lot of farms are bringing in more like 3 to 10. So pretty high density. Does that answer your question?
Yes, ma'am. I just was curious, more so like for organic production, the honeybee population and foraging, did you see any difference between that and conventional side of it, or was there a study done on that? As far as number of bees, even though, we might bring in 5 hives to the acre as far as honeybees in organic production, but that same conventional grower may have the same 5 hives. Was there any difference just because of products or insecticides being used, or any pheromones that are in the field that that may deter that honeybee?
That may deter that. Yeah, that's a great question. So we actually did find that when you look at the number of bees in the crop row, it was slightly higher on the conventional compared to the organic farms. I think that might be because of the number and diversity of plants that are competing with blueberries for bloom. So in organics, they might have either more flowering weeds or more natural habitat adjacent to the farm. So if anything, we actually found that organic farms were getting slightly fewer honeybee visits for the number of hives they were bringing in compared to the conventional. And that's why I mentioned that plants that are competing with blueberries for bloom, we think is also an important factor. And there may be more plants competing with blueberries for bloom on organic farms, just because of the diversity of plants both within the farm and adjacent.
Makes all sense.
Yeah.
Thank you.
Yep.
Thank you so much.
Yeah.
Oh, that's so kind. Definitely, yeah. We can continue with the questions after the session. We thank you so much, Dr. Malinger, for your practical speak. It's great information for the growers. Our next speaker is Jonathan Oliver from the University of Georgia.
Thank you. Yes, I am Jonathan Oliver from the University of Georgia, and I am the fruit pathologist and extension specialist based out of Tifton. And today I'm going to give you an update on a Georgia blueberry pathology update. So for my talk, I'll talk about some of the disease issues we saw on blueberries this year, this past season. I'll also talk a little more specifically about 3 types of diseases, algal stem blotch, fruit rots, and leaf rust. And then I'll end by talking about the seasonal spray schedule for commercial blueberry production. So first, what did we see most often on blueberries during the 2023 growing season? Well, based upon samples submitted to the Plant Disease Clinic in Athens at the University of Georgia, we diagnosed 172 issues on blueberries during the season. The most prevalent issues appear to be root diseases. We see that really every year. Over half of our submitted samples are affected by Phytophthora or Rhizoctonia. We also this year had, kind of unusually, some issues with algal stem blotch that showed up in several locations, and I'll talk about those a little more in a moment. We also routinely see a lot of issues with blueberry leaf rust as well as anthracnose as well. So that sets up today's talk. So for algal stem blotch, this is actually a disease caused by an algae, which makes it kind of unusual and unique. The algal species that causes algal stem blotch, Cephalurus virescens, also infects other plant hosts, including blackberry, magnolia, and camellia in the southeastern U.S. And if you saw my talk during the caneberry session earlier this week, orange cane blotch caused by the same species on blackberry is a significant issue as well. But previously on blueberries anyway, in Georgia, algal stem blotch had only been observed sporadically, and it was not causing major issues. However, for the last several years, it's been known as a significant issue in southern highbush production in Florida, and it appears that this issue may be creeping northward somewhat. In 2021, we kind of had our first, I guess, explosion of issues with algal stem blotch in Georgia. That was a very wet year, and we had a lot of growers report issues in the fall of that year. But also this past season, we had several sites report problems with algal stem blotch, including some more significant issues than I had seen in Georgia previously, where plants were literally dying as a result of algal stem blotch. So the symptoms of algal stem blotch, the algae actually grows beneath the cuticle of the stem, and it causes blotches that become more conspicuous once the algae produces these sporangiophores that stick through the cuticle of the plant, and that's why it's called an orange, it's a algal stem blotch. The leaves on the affected stems are also chlorotic or bleached white, and they have kind of this odd kind of speckled appearance, and I've never seen this kind of chlorosis with anything else besides algal stem blotch. It almost looks like somebody splashed bleach on the plant. It's very strange. But the leaves can also bleach completely, yellow and be of kind of vibrant yellow color. Plants that are severely affected lack vigor, and they fail to regrow after summer pruning or hedging, and the affected stems can defoliate, and we saw that more than we've ever seen it this year. I'm not sure if you can see how well you can see it in this picture, but there's multiple stems here that are completely defoliated as a result of severe issues with algal stem blotch, and the leaves that are there are very chlorotic, and they're going to fall off soon. So very limited information exists regarding control of algal stem blotch. Some practices from the cultural side of things that may reduce disease spread include utilizing disease-free stock for new plantings. Anything that can be done to reduce plant stress could potentially reduce issues with algal stem blotch. Any infected canes can be removed and destroyed. Overhead irrigation, this is an algae, so of course moisture is essential to its life cycle. Overhead irrigation can possibly contribute to issues with algal stem blotch, and anything that can reduce the amount of moisture in the canopy. So if you have weeds growing up, pretty high within your blueberry plants, that can hold moisture, that can keep it moist in there, and that will potentially allow for the algae to spread and reproduce more easily. From the chemical control side of things, we're kind of limited because this is an algae and not a fungus, so most fungicides are not going to be effective against this organism. But copper fungicides, including Coside 3000, are recommended and do reduce algal sporulation and protect the healthy canes. So these types of copper products are not going to be eradicative. They're not going to clean up the issues you have with algal stem blotch, but they can protect the stems that are not affected and the plants that are not affected. And so there's a Section 2EE label for use of Coside 3000O for algal stem blotch, and it recommends monthly applications after harvest. But if you want to know more information about algal stem blotch, I have great news. This afternoon at 2:40, right in this room, Dr. Phil Harmon from the University of Florida, who has a lot more experience with this based on their issues in Florida, is going to give an entire presentation on the biology and management of algal stem blotch spreading northward. So look forward to that if you've had issues with algal stem blotch. I'm going to move on and spend a good chunk of time talking about some fruit rots, especially some findings from our lab of fungicide resistance among the pathogens causing fruit rots on blueberry, and some field trial results from some work we've done over the past season. So there's a lot of different fungal organisms that can cause rots on blueberry, and some of those are shown here, but the three fungal diseases fungal rots on blueberry that cause issues most routinely in blueberry production are anthracnose rot, alternaria rot, and botrytis gray mold. So anthracnose fruit rot or ripe rot is caused by a couple of different species in the genus Calotoctracum. In the case of this disease, fruit infections begin as early as bloom, and they often remain latent until the fruit ripens. So the fungus can infect, when there's flowers out there, or even when the fruit is just beginning to develop, but the fungus will essentially stay there and do nothing that we can tell until after the sugar levels begin to rise, and then that pathogen will grow rapidly within that berry and cause it to shrivel, potentially in the field, and you can sometimes see this orange sporulation from this fungal organism on the surface, and these sticky spore masses that look orange are problematic because, of course, that's how the disease spreads to other nearby fruit. Anything that touches those sticky spore masses, any other fruit can acquire the disease, and of course, those fruit, once they're picked, they touch they touch all your surfaces on your packing line, in your lugs, clamshells. The disease can spread from there as well, and so that's why this can be a major issue post-harvest also. Warm weather during bloom and just before harvest is associated with more problems with anthracnose ripe rot. Alternaria fruit rot is another important rot of blueberry, and it's a fairly similar story. You'll often see it as more of a gray-green mycelium on the surface of the berry. Often you'll see it most associated with that stem scar end of the berry. So if you pick the fruit, and especially if there's a wet stem scar, sometimes the alternaria will sporulate out of that and grow from that point. And unlike with anthracnose, cooler and wetter conditions favor disease development with alternaria. So warm weather with anthracnose, cooler weather with alternaria, depending on when in development, when along the season, and what your weather are you have at your particular location, you may see more issues in a given year with alternaria or anthracnose. Botrytis gray mold is another important fruit rot. You've probably seen, if you've bought blueberries from the store and left them in your fridge too long, eventually you'll see these fuzzy, gray berries, especially, and it also causes issues on strawberries, and you may have seen it there. But actually on blueberry, we think most of the losses caused by botrytis are actually when it affects the blossoms. So before fruit can ever even develop from these blooms, this pathogen can blight those blossoms. Wet conditions favor issues with botrytis. So if you have wet conditions, especially around bloom, you can have significant losses due to botrytis. And botrytis is a necrotrophic pathogen. It likes dead tissue. And so whenever there's dead tissue out there, it can reproduce on that dead tissue, it can sporulate, and those spores can blow to nearby blossoms and infect. And so if we get freeze damage or frost damage around bloom, in those years we can have more significant issues with botrytis than in other years. We don't always recommend fungicide applications specifically targeting botrytis, except if we had some of those predisposing conditions like a late freeze or a freeze during bloom. And in those cases, we definitely would recommend chemical applications in commercial production. So, but talking about fungicides, all of these pathogens, unfortunately, are very good at developing resistance to the fungicides we use in blueberry production. And for anthracnose, the Calotoctracum, it has already developed resistance, and we know that from work we did back in 2019 to both components of the fungicide Pristine. A grower there near Blackshear, Georgia, was reporting major issues with anthracnose. He'd been on a pretty good spray program. We went out, we collected isolates, and we found that they were resistant to some of the products he'd been using, like Pristine and Abound, which in the past had been very effective for fruit rot control. So that was a major concern. So during the 2021 and 2022 growing seasons, we did a survey of fruit rots from blueberry in Georgia. We collected fruit from 46 different sites in the 6 major blueberry producing counties in Georgia, from both rabbit eye and southern high bush, and we collected that fruit, we isolated, we brought those isolates back to the lab, and we looked to determine how prevalent fungicide resistance may be among those pathogens. So among the Alternaria and Botrytis and Calotocricum, we screened these for resistance to common fungicides, and we did this using a mycelial growth inhibition assay. Basically, we had media where we grew the isolate on. So the top row here is one isolate, the bottom row is another isolate, and as we increased the concentration of fungicide in the media, we saw that some of our isolates, basically stopped growing on the fungicide-emitted media. Other isolates grew just fine, and so this isolate would be an example of one that has resistance to this fungicide. So we did this for a lot of different isolates in the lab, 46 Alternaria isolates, 117 Botrytis isolates, and 50 Calotocricum isolates were tested, and we screened these isolates against a large number of active fungicide ingredients that are used in blueberry production. What we saw for looking down the column here for Alternaria was that most of our Alternaria isolates remain sensitive to most of our fungicides. We did find some isolates, a percentage, 22% of the isolates tested and 46% respectively, that had resistance to pyraclostrobin and boscalid, the 2 components in the fungicide Pristine. For Botrytis, it's very good at developing resistance to fungicides. We saw a fairly low level of resistance, between 6 and 10% for most of these fungicides of our isolates had developed resistance. Of note here, across the board, the active ingredient fluazinam, which is in the fungicide Omega, we did not find any isolates of either Alternaria, Botrytis, or Calotocricum to be resistant to this fungicide. For Calotocricum, we found that 18% of our isolates were resistant to pyraclostrobin, and 36% of our isolates were resistant to fludioxonil. And this is a major concern because Calotocricum isolates have previously been shown, and in our testing they were, insensitive to SDHI-type fungicides as well as saprodinil. And so even though Pristine and Miravis Prime and Switch are combination fungicides that include 2 active ingredients, at least one of their 2 active ingredients is not going to be effective against Calotocricum. Therefore, if resistance develops to the other active ingredient, those fungicides theoretically would not be effective at killing those particular isolates. And if the proportion of resistant isolates in your field is high enough, then you may begin to experience control failures when utilizing these fungicides. Looking across all of the different testing we did, 13% of our Alternaria isolates were resistant to both components of Pristine. 18% of our Calotocricum isolates were resistant to both components of Pristine. 36% of our isolates from 10 out of the 16 locations where we had collected isolates that we tested, these were resistant to all the components of both Switch and Miravis Prime, and 14% of our isolates in 4 of these 16 locations were resistant to Pristine, Switch, and Miravis Prime. And obviously that can be a major concern because those are some of our frontline materials for rot management on blueberry. So what can we conclude from our work so far? We definitely have resistance in Alternaria isolates to the QOI fungicides and boscalid, and we have Botrytis and Calotocricum isolates with multiple fungicide resistances in Georgia blueberries in at least some locations. To ensure that these fungicides remain effective against fruit rots, it's more essential now than ever to utilize good fungicide resistance management practices. So what do those practices look like? It's really important to rotate our available modes of action, not to rely specifically on 1 or 2 modes of action throughout the season. And that will prevent or reduce the selection pressure on the fungi to develop resistance to those fungicides specifically. Also, as we've recommended for years, tank mixing with multi-site fungicides like Captan or Zyram can reduce resistance development. Multi-site fungicides are much less likely to, based on their activity, are much less likely to lead to resistance development. And so if we can tank mix things like Switch with Captan, we're less likely to lose efficacy in Switch in the long term because resistance will not be as prevalent and will not develop as easily. We did find no resistance among our isolates to the active ingredient in omega, fluazinam. And so as such, as we've seen in several of our trials over the last few years, omega may be a good rotation option for fruit rot control. Likely it's best utilized around petal fall. So it has a 30-day pre-harvest interval. It's rates a good or fair against these fruit rots. So it may not be what you want to use right at bloom when you probably want to use some of these more excellently rated materials like Switch, Pristine, Marabas Prime. But instead of maybe spraying Switch twice, once at bloom and once at petal fall, maybe substituting a spray of omega at petal fall might be a good rotation option for at least some of our growers, especially if you have experienced control failures or you want to head that potential problem off. All of our alternaria and Colletotrichum isolates were also sensitive to the DMI fungicide quash. DMI fungicides are typically effective, and we think about those when we think about control of mummyberry and when we think about control of rust. And so if we're using quash during the bloom to harvest period, it may also be an effective product for fruit rot management as well, and we don't have resistance to it at this time. And we also conducted some trial work this past season to compare, we conducted multi-site trials to utilizing the Blueberry Advisory System, which is available from AgriClimate. This system has been hooked up and utilized for several years in Florida, but in 2022, the 16 UGA Weather Network stations got hooked up to this system for the first time. And so this system, in brief, predicts the risk of new anthracnose infections based on weather data. And so what we wanted to do was compare kind of a standard program for fruit rot management to a program informed by the Blueberry Advisory System results. And we compared these to an untreated control. So our standard program that we've utilized in our trial work for the last few years includes 5 sprays starting at 10% bloom with an application of Switch, a petal fall application, an application 10 days after petal fall, 3 weeks after petal fall, and a pre-harvest application. For the Blueberry Advisory System program, we only sprayed as needed. So basically, whenever the advisory system gave a moderate or high risk of infections occurring, we would apply a fungicide, and we used fungicides rated in the Blueberry IPM guide as excellent, like Marabas Prime or Switch, when there was a high risk event, and good materials like Omega or Captan for moderate risk events. So at all 7 locations that we conducted this trial, we saw a numerically less anthracnose fruit rot and all rots versus the untreated control. So I color-coded this for ease here, but basically the highest level of disease of each type of disease we saw is shown in red. Green is no disease, and shades of light green, yellow, and orange are in between those 2 values for each location. And you can see that across the board, we saw less anthracnose fruit rot and all rots at all 7 locations. We did not, however, see a major difference between the 2 programs, the standard program or the Blueberry Advisory System program, except maybe at one of our locations where we had very high amounts of anthracnose. So we saw 27% of our fruit at this Nehunta rabbit eye site were affected by anthracnose fruit rot. And there we begin to see a difference between the 2 programs, and the Blueberry Advisory System program performed better in that case, dropping that down to only 1% of the berries being affected by anthracnose fruit rot. Both programs generally resulted in the same number of sprays, even though we only applied fungicides if the advisory system gave us an alert for high or moderate risk of infection in this particular season. In other seasons where maybe conditions would be less good for anthracnose, maybe we'd spray less using the Blueberry Advisory System, or maybe we'd have sprayed more if we had a really wet and warm year in the spring. But we generally sprayed all the sites approximately the same number of times. However, we did find that using Captan or Omega, so these good efficacy products alone during moderate risk events, actually, since they're a little bit cheaper than some of these other fungicides, actually resulted in the spray program on a cost per acre basis being cheaper in 5 out of the 7 locations. So based on our estimation of the cost of the program. I think only in one location was it actually more under the Blueberry Advisory System program, and that's because the advisory system there recommended an additional spray over the other program. But all of the other cases, it was either lower or the same. So I want to talk briefly about leaf rust. So blueberry leaf rust is caused by the fungus Pucciniastrum vaccinii, and it overwinters on plant debris. And so it overwinters, it's usually on, we think, on leaves that basically hang on to the plant throughout the winter. So we have some winters where it gets cold enough, the plants defoliate. We may not see issues with rust as early in the season, but in other years, it gets, our rabbit and our high bush can hang on to their leaves all winter. It can start that rust epidemic, it seems like earlier and earlier every spring, and we're seeing increasing issues with leaf rust in Georgia. But how you can recognize leaf rust, if you look at the top of the leaf, you'll see these either necrotic or kind of dark, maybe purple colored spots. Thank you. And if you look directly below that, flip the leaf upside down, you can see an orange pustule, and that is a rust pustule, and so it can be easy to identify if you flip the leaf up. And it can prematurely defoliate plants by late part of the season if it's not controlled in some cases. We rarely see issues, but we can see issues with rust actually on the fruit. These will be pustules on the calyx end of the blueberry fruit in severe cases. The DMI fungicides, as I mentioned before, are typically what we use for control of rust in blueberries. But we wanted to conduct some additional trial work in 2023 to look at some other materials, some conventional products, as well as some other organic fungicides for rust management. And we conducted this trial on southern highbush farthing at the Blueberry Research Farm in Alma, Georgia. So I want to show you those results briefly. I've color-coded it again, more disease is red, less disease is green. But we looked at 4 OMRI-listed fungicides that are labeled for use in blueberry, as well as 5 conventional products that are some of which are labeled currently for use in blueberry. The organic products were sprayed approximately every 7 to 10 days, so we made 10 applications starting on July 7th. For the conventional products, we only sprayed those basically every other time, so we sprayed those 5 times across the season. And what became pretty obvious from our data is that we got great control of rust, so we reduced both the incidence and the maximum severity, as well as the area of the leaf affected by pustules dramatically when we utilized products like Sevia, which is a DMI fungicide from BASF that has recently been labeled for use on blueberry. Also, chlorothalonil or Bravo reduced rust issues dramatically. And both rates we used of a new fungicide, Alesis, which is a DMI-SDHI combination product from BASF that they're in the process of seeking a label for blueberry. And so all of these products were very effective. Among the organic products, you can see that they were relatively less effective, though some of them did have some efficacy against rust. Among these, Howler Evo was the most effective organic product in our trials. Last, I'd like to leave you with a seasonal spray schedule for blueberry. I throw this example schedule up every year, and so how might it have changed from one year to the other? Well, first, let me explain it. So from the late dormant through after harvest in a given season, we have multiple disease issues we're generally contending with in Georgia in commercial production. We have Exobasidium that we begin to be concerned about fairly early in the season. As we move towards bloom, we have a variety of diseases, mummyberry, twig blight, fruit rots, and botrytis that are of concern. And as we move later into the year and towards after harvest time, leaf spots become our primary concern. And so to control all of those diseases, if you're dealing with all of those simultaneously, the spray program can be pretty intensive and require multiple fungicide applications across the season. I've color-coded some potential fungicides which would target the diseases that are prevalent or important to control at that time. So you see that generally an application of sulfurix in the late dormant period can take care of Exobasidium issues. Around green tip and into bloom, mummyberry and twig blight should be controlled often through applications of things like DMI fungicides. Something like switch can target fruit rots and botrytis if applied around bloom. Around petal fall, I've put in a spray of Omega and Captan, which may be a good rotation out of a progression of switch or Moravis prime. And we often are seeing rust issues pretty early on, so in the cover sprays we have to think about applying things like DMIs to keep rust down. And as we move towards the after harvest time, then we're only concerned really about leaf spots. And so Prophyte and K-Phyte can target things like septoria. The DMIs can target rust pretty effectively, and Bravo can take care of both types of leaf spots at that time. A lot of this fungicide efficacy information that I've included today is reflected in the Southeast Regional Blueberry IPM Guide. And that IPM guide, the 2023 version is what's online now, but the 2024 version should be put up pretty soon. Within that guide, there are various tables that give information about the types of diseases that are important at different times across the season, and a list of fungicides that may be used to control those. A lot of the pictures that I showed today to help you recognize these diseases, as well as the fungicide efficacy information that I've talked about, is available through the My IPM app. And this app is freely available for download through the App Store or Google Play. And if I have any time left at all, I'd be happy to take a question or two. Excellent. Any questions? Yes, sir.
Does Omega have any activity on rust?
We do not currently recommend it for rust. I don't know that I've specifically looked at it for rust, but I don't know if it would have activity on rust. I can't say that from any experience. We don't rate it as having any efficacy in the guide currently for rust. Yes, sir.
Are you seeing any problems with Neopestalotiopsis post-harvest?
In like causing rots on blueberry?
And Neopestalotiopsis.
But you mean, when you say after harvest, you mean on the fruit?
Yes.
We have identified Neopestalotiopsis on fruit. I actually have a student looking to see if it's actually causing a rot or if it's just something that is also present. So Neopestalotiopsis species are very common. They're a common find in the field. They actually overgrow isolation media pretty easily. So we do isolate it pretty frequently from blueberry fruit, but we don't know whether it's actually causing any significant issues on blueberry or not. But we are looking into that just to see. But yeah. Yes. Okay.
I think it was that harvest you might have talked on.
Mm-hmm.
You had just got done on the bound or steam or, having issues with resistance. So are you saying that you add Captan that you're going to be good, or is that just an old slide?
So if you have resistance to Abound at your location, it would not be providing the control you're hoping to achieve with it. We definitely didn't find resistance to the QOIs at every location we looked at, and so it still could be a useful product. It has broad spectrum efficacy against a lot of different fungal organisms, including ones I did not list among these that are causing fruit rot. And so, yes, while it is, I guess, an older recommendation, it is still a potential product to be used around right before harvest. Moravis Prime also is something we've used at that timing, and so other products could be used for sure. So. All right. Thank you.
Thank you so much, Dr. Oliver. I just want to remind that all the recorded sessions will be made available to all registered attendees after the conference. Our next speaker is Dr. Ash Syal.
Good morning. My name is Ash Syal. I am a blueberry entomologist at University of Georgia. And today I'm going to give you an update on insect pests of blueberries in the southeastern US. I'll start off with this table that shows sort of whole season of blueberries, blueberry insect pests at a glance. These insect pests are ordered in by the time of the year or season in blueberry crop. Starting out with the bud mites, they start with the pre-bloom and basically post-harvest issue primarily. They get into pre-bloom in some situations, but primarily post-harvest issues, scales and borers post-harvest, gall midge and thrips. They start in pre-bloom period, get through bloom into mid-season. Aphids, leafhoppers, they are primarily starting in blueberry bloom period, get through mid-season into pre-harvest. Then we get into, as fruit development starts, we start to get into this fruit worm issues, cranberry fruit worm, cherry fruit worm, and there is a lot more species in the southeastern US that you may come across. Some leaf, generally referred to as leaf rollers or leaf tires. So you will see lots of those species. These two species are more common. Plum curculio, again, is starting with bloom through mid-season into pre-harvest. Then we get into the major ones when our fruit starts to ripen is blueberry maggot, which we have not recently seen as much in the southeastern US. It is still a key issue in some other regions in the US, but we have not seen in the southeastern US lately But spotted wing Drosophila is a key pest in blueberries. As soon as we start to see those blueberries change color, that is when susceptibility window starts, and then it continues through end of the harvest. Then we have white grubs and ground pulse. They are again occasional problems in some situations. But this is sort of a general list. We may have a lot more pests that you may see in blueberry systems. Today, I will cover only a select issue of this list. I will not cover every single pest. I will cover only those that I have received several reports from either farmers or extension agents. And if you have any issue that I do not cover, please feel free to ask me after the presentation, or just stop me somewhere around the conference. I'll be around until after lunch today. So blueberry bud mites is a really serious issue, or can be really serious issue in some locations. We have seen it occasionally everywhere, but a couple years ago, we saw a situation where there was 100% crop loss. It went to that extent. They are really small, tiny areophyid mites. They are not real mites even, because mites do have 4 pairs of legs. They do have only 2 pairs of legs, the right kind of front part of the body. As you see in this middle picture, it's like a banana-shaped structure. They're transparent whitish color, about 1/128th of an inch, really small, which is approximately 200 microns body size. Females lay up to 200 eggs. They develop through 4 stages in about 15 days. So they can turn over generation relatively quickly. They are really small. They can disperse primarily by air when they are out on the plant tissue. They can get airborne and can move long distances, in fact. They can also be dispersed by crawling or hitchhiking with either other plant material or farm workers in the farm, in those fields. Populations peak in December through February and decline in summer due to high temperatures. Mild winters tend to boost population. That is what we have seen as a problem. The year we saw extreme issues, that was a direct result of the mild winters we had. These populations, they actually start right after harvest. They find the birds that are going to make flower and fruit birds next year. They get into those birds and basically spend winter over there. They feed while within the birds and then transfer toxins along the side. And those toxins cause the symptoms that you see in this picture here, and just sort of blisters and those kind of pimple type symptoms, which are very distinct symptoms for bud mites. As a result, they can desiccate and the birds fail to open. They kind of tend to form this bushy shape that you see in these pictures. Flowers and berries develop from infested birds usually have small blisters and pimples, as I mentioned. Summer generations cause retarded leaf and vegetative growth, which negatively affects next year's crop. So we see the impact the following year, and that's what we did in this particular situation. Sampling for bud mites is relatively easy. You just collect branches with approximately 10 branches from a field with 5 buds on each. You can look them once you have 50 birds. You look under microscope. If you have 10% of those infested, that may be time to start thinking about making application. Controlling is relatively easy of bud mites, but the problem is they're always inside the buds and they're very small. It's very hard to find them. So you have to use either microscope or hand lens, or have to have really trained eyes to look for symptoms and track them. If you do have infestation or suspect infestation, post-harvest pruning is one of the best ways to get rid of them, because they are not very mobile. They're only exposed when they are moving from one bud to another, when they're looking for fresh buds. But otherwise, they are inside the buds. So if you prune the buds, it's one of the best ways to get rid of them. We do have lots of miticide and insecticide options that can be effective. Portal, Oberon, acromide, brigade, danitol, savin, abamectin, movento, and also oils is most common way to control those. One issue, as I mentioned earlier, they're always inside the buds. They're sort of protected from spray residues. So the best way to control them is to spray really high volumes and also use high pressures so the spray residues can get into the bud folds to get to where these bud mites are feeding. You can also add surfactants with the spray mix to make sure that whatever spray residues get on the plant tissue, they actually stick there for a while and maybe get in direct contact with the mites when they are exposed. So again, control is not an issue as long as you make the spray at the right volume and at the right time. Now, spider mites are real mites. There are several species of spider mites that can be a problem in the southeastern U.S., primarily in Georgia and Florida. Southern red mite is one of the common species that we come across. They can also cause real serious problems. They're also known as web spinning mites because they do have some webbing wherever these pests occur. They're primarily immediately post-harvest type timing issue. That is when some farms tend to ignore the bushes. And that is when, in terms of weather or season, we do get some dry period as well. When bushes are otherwise compromised or not well irrigated, these types of secondary issues, they do take over during those opportune times when blueberry bushes are otherwise compromised due to some either nutrient or irrigation issues. Symptoms include leaf bronzing, which is very characteristic. You can, if you have high populations, you can see it from a distance, and they can cause serious problems in terms of compromising the bush's ability to photosynthesize and, of course, leading to serious issues with the quality of the fruit and yield the next year. They can build high populations in a relatively short period of time because their life cycle is also very short. We do have lots of good options when it comes to controlling them. Miticides like Portal, Oberon, and Acromite, they are the best option to go with. And these miticide, again, we did not have these miticide options until maybe 2 or 3 years ago. These are most recently registered products and still do have a good efficacy against these mites. Another issue that we have come across more often and scales. Scales have historically been a problem, sort of a sporadic issue here and there, but more recently it has become really widespread issue, and almost every farm has scales to some extent. Now, Southeast has a very unique situation where we have several species of scales that can be problematic. At any given location, it's sort of a complex of several species, and those species can coexist at any one location. So if you see the one characteristic species at one location, at other location there may be another one, or it could be a mix of several species. And as you see in this picture in the background, this is sort of a typical high pressure situation where we do have several layers of scales on this bush, and it goes all the way from really the ground level up to the top of the canopy. They can feed on fresh plant tissue. They can actually feed inside under the bark on canes, and can cause serious problem with the bushes, even leading to death of the bushes if left uncontrolled. More recently, we have seen mealybugs, which are also very closely related species or group rather, to scales. They are sort of close cousins of scales. The only difference between scales and mealybugs is that when their eggs hatch, the first stage is called crawlers in both cases. Crawlers are the ones that are really tiny critters. They just lots of them, and they move really fast in search of finding a spot to establish their mouth parts to start feeding. And once they establish their mouth parts and start feeding, they mold to the next instar, second instar, And at that point, scales, they lose their legs. Scales do not have legs beyond that point. Once they establish their mouth parts, that is where, and molt, they will basically spend the rest of their life at that point, at that site. Whereas mealybugs, they retain their legs. They can feed at one location, and if they're not happy, or disturbed, they can move to another location because they do retain legs. So that is the difference when it comes to identifying between scales and mealybugs. But otherwise, they are very closely related and do suck the juice out of plant tissue. They feed the same way and cause same problems in bushes. As far as control, we do have a number of options. Again, this is one problem that is really complicated to control. Once you have high populations established, it takes a number of years to actually control them completely. It may take 2 to 3 years to control because they can survive in really small, tiny cracks and crevices of the plants or even soil, and then keep establishing new colonies at different locations. Because those crawler stage, they can get windborne and actually travel long distances once they are windborne, or can also hitchhike and move from one location to another within one field or in between different fields. So it's a very complicated issue to control. You want to make sure as soon as you detect infestations, you want to stay on top of those with one of these applications. Oil applications are extremely important, but with oils you have to make sure that you use high spray volumes and high pressures to get good coverage. If you don't get good coverage, oils will not help. Other insecticides that we have as options, Sivanto, Movento, Admire, Assail, or even organophosphate applications at the crawler stage is the best way to go. Another strategy to have a good control of scales is to keep monitoring for crawlers, because crawlers is the most susceptible stage to any of the insecticides. If you can make the good timing, this will be easy to control. Historically, we have made scale applications in the fall, or so maybe during the dormant period with oils. We compared some of the insecticides and oils during the fall in November or dormant stage, and during post-harvest in July, August. Same products gave better control during the post-harvest timing, and that's what we are recommending now. The reason is that at that point, the plant cell sap is more active, and scales are also feeding more actively. It means they can pick up residues more effectively, and it can impacted by the residue that we spray. As a result, we get better control during that time. Flathead borers is also a sort of a secondary but an opportune issue in blueberries. There are occasional pests at some locations. They're more in Florida than Georgia. We did a survey. We found maybe 1 or 2 sites, but not much. In Florida, I have heard that they're more common. Adults are beautifully marked, metallic colored beetles, about 1/2 inch long. They have short antennae and long, very large conspicuous eyes. It's very easy to identify. Damage occurs when larvae bore into the canes, and basically from the outside they look like small hole, as you see in this picture here. But inside it's really basically a big tunnel that they bore and can cause a lot of damage. They can actually lead to killing the bush. So prevention of the bushes from mechanical damage and wounds is, because I mentioned, they are opportune problem when blueberry bush health is compromised. Otherwise, especially due to lack of irrigation, these issues do become problem. These pests become more of a serious issue there, or if bushes are stressed for some other reasons. So make sure when you use equipment with in the field, just try to avoid causing physical injury to the bushes, and also keep them well irrigated so they don't otherwise get compromised. Once you have an infestation that you detect in the field, it's one best thing you can do is to prune the infested bushes. In that particular field, do a bit more heavy pruning as compared to what you would do normally, just to get rid of any of the infested canes that may be present. Then once you prune, remove those canes out of the field, either burn them or bury them really deep to make sure you don't leave the source for reinfestation in the field. So yeah, if you still want to, if you do prune and still have infestation issues, there are some insecticides that you can apply, and admire through drip irrigation is the best way to control these. Blueberry gall midge, again, is a serious issue. It's everywhere in the southeast and can cause serious problems. Females of these are tiny flies with really long legs. Females lay eggs in flowers and vegetative buds starting stage 2. And they feed inside the buds, can basically kill those buds. Up to 80% flower bud loss has been reported in southeastern U.S., in Florida and Georgia. Midge injury is easily underestimated because of the timing. Especially in Georgia, we can confuse that injury with the cold injury, because cold injury also from this in the middle of the bush does look like the same sort of a dark spot type symptoms. Monitoring for gall midge is extremely easy. You just need to collect flower birds a couple of times a week, put them in Ziploc bags, and those little larvae, they're really small, but they're easily, you can see them without any hand lens or anything with the naked eyes. They automatically come out. You don't need to do anything. Just put them in Ziploc bag. And then if you have, if you see those larvae out there, and you can also use double sticky cards like in this figure, or also inverted buckets. These two methods are recommended for adult monitoring. May not be economical in some situations, but that's one way to monitor adult gall midge. Controlling gall midge, another good way that we have developed in Georgia, it may work in Florida and other situations as well, is to see if there is any way to be proactive in estimating what type of populations and when we may see those peak populations. We sampled fungus gnats, which are also related to gall midge. And what we noticed is that fungus gnats peak about 2 to 3 weeks before gall midge peaks. So this gives a sort of about a 2 to 3 week time frame to prepare for these gall midge applications if you have a history of infestation at your blueberry field. So that, and also because they feed inside the buds, these gall midge applications are always a proactive sort of management, because if they're inside the bud, insecticide application is not going to help. Here is a guide that we developed to help you identify between fungus gnats and gall midge. So if you see too many veins, few or no hairs on the wings, and spurs present on the legs, then that is not gall midge. Gall midge have very few veins, just one full straight vein and one that bifurcates toward the end. They have lots of hair on the wings and margins and do not have spurs on the legs. Easy to identify. So control. So flower bud, stage 2 is where we need to make sure we protect birds starting from stage 2 onward, because that is when females can lay eggs. And we want to protect those birds before females lay eggs. Diazinon has been our key tool. Unfortunately, this year we are having some problems due to manufacturing issues, that diazinon is not available in the market right now, or at least it's short. But luckily, we do have some other products that are effective and can be used. Delegate, Entrust, Sale, Imidan, Sivanto, and Movento are other options that can be used to control gall midge. As I mentioned, midge insecticide applications are protectants, because we want to make sure they are applied before females lay eggs to protect those birds from larvae getting inside them. So timing it will be the key, and that sampling does help with that. Flower thrips is the next issue, and this is one of the most complicated issues in Southeast because we do have several species of flower thrips. And more recently, we have gotten another issue, chili thrips, which is a sort of a different timing. Even though it's thrips, it is not this timing. So I will discuss that later. Flower thrips are a complicated issue in many ways. They can cause problems in, they feed on leaf and flower surfaces. They're active before, during, and after the bloom. And that is what complicates the management of flower thrips. We will see that these feed on both flowers and also fruit. You can see symptoms on both, as you see in these sets of pictures. These are symptoms we see in flowers, and these are the symptoms we see when they have the thrips injury in on the fruit. Damage in southern highbush can be up to 60% in this in extreme thrips infestations. Primarily in the southeast, flower thrips are a rabbit eye problem, as you see from these studies that one of my grad students did. During high bush period, we did not see high populations. It's primarily rabbit eye problem. Monitoring, again, is very easy. You just need to collect blooms 2 to 3 times a week, put them in Ziploc bags, and thrips will automatically come out of those. You can get an idea of how many thrips you have, and if the numbers are more than 2 thrips per cluster of 8 flowers, that is when you need to start thinking about making applications. Control of thrips is challenging, as I mentioned earlier, because of the timing, because it's bloom time, and we want to see lots of good bugs like pollinators that Rachel talked about in the field thriving during that time. So we want to be very careful on what we spray and how we spray. Diazinon, of course, if it has not been used for gallmage and is available, this is one of the good tools, but we need to make sure we make either early morning or late evening applications to minimize exposure of the pollinators. We do have other insecticides like Entrust, of course, is for the organic farms. Delegate, Assail, and Sivanto are the other options that may work well for flower thrips. One of my grad students is now working on thrips management. His studies showed that Assail was effective. There were several products effective, but Assail was the best out of all for southern highbush time period, and Assail and Delegate were the most effective during the rabbit eye timing. So these are the products that we are highly recommending when it comes to that. And we also compared 1 spray application versus 2. And as you see, when you make 2 spray applications about 7 to 8 days apart, you get full bloom coverage, full bloom time completely covered if you have high populations, and if 1 spray controls, but then you have a reinfestation, 2 sprays may be best to get to the whole entire bloom time. Chili thrips is a new invasive species I mentioned. They're smaller in size than the flower thrips, but they can be more injurious than flower thrips. The good thing is their timing is different, so there's a less chance that you will get confused. But in Florida, it's everywhere in blueberries, and I have heard recently that they have to make 9 to 12 spray applications during post-harvest time just to control chili thrips. In Georgia, these counties have been already reported to have chili thrips, and it is basically on the move. There may be more counties during this coming year and onwards. Here are the symptoms that these chili thrips can cause. They are primarily foliage feeders. They suck the sap out of plants and cause these symptoms on fresh, lush of growth that you see during the post-harvest time. That is when they can cause these symptoms, bronzing and darkening of the tissue, leading to even death of the bushes. I heard from some of the Florida experiences. These are other symptoms that can overall compromise the vigor and health of the bushes. These are the products that Oscar Leiber's program tested on chili thrips, and this is what they showed, that most of these products were effective, but Assail and Apta were the most effective. And I also learned earlier this week that some farmers are using this insect growth regulator, Ryman. They are basically dosing it several times during that chili thrips time to get through the chili thrips season. Now comes our key problem, spotted wing Drosophila. By now, everybody knows that this is a serious problem across the United States. Everywhere there are small fruits, SWD exists. Females have this ovipositor, unique sclerotized ovipositor they use to deposit eggs. Males have these dark spots to really easily identify themselves from other similar flies. Once females lay eggs, those develop very quickly, turn to complete the cycle from egg to adult in 8 to 10 days. That means they can turn over many generations during one season and cause a lot of damage and huge impact on blueberries and other small fruits nationally. In Southeast, this is how their seasonal phenology looks like. They basically are around year-round. You can trap them. And also, they're not only in the field, but also around the field and the wooded areas as well. That means we have them here to stay and to cause problems year-round. Here are the major non-target plants that they can develop and wait until the blueberries are back in the field to cause more problems. This is the data from one of my grad students' work showing that. One thing you can do is maybe do burning. We have noticed that this does help temporarily with some of the SWD populations. Sampling for SWD has traditionally been done using liquid, stinky, very inconvenient traps, but over the years we have done a lot of work and developed this very convenient red sticky card-based trapping system, which is basically lured with one of these commercially available lures, and it goes, one lure can last for 6 to 8 weeks. Not much changing. You just need to go back to the sticky cards to see every week to get the idea of how many flies do you have or what population levels you have. Otherwise, it's really easy to use this method to sample. And it's just as good or even better than those liquid sticky traps. Now my postdoc is actually developing a larval sampling method as well. Historically, what we have used is salt extraction and sugar extraction, where we had to do a lot of work to prepare solutions and then boil them and then crush them and all that, which is very inconvenient. And he has developed this method where we use this suction method. Basically, if you have a vacuum chamber, vacuum method can be very conveniently used to sample for any larvae that may be present in the blueberries, and it's very convenient as compared to other methods, and is just as good or even better in some situations to sample for larvae that are inside the fruit. Biological control is another method that we are focusing on, SWD control. After 10 years of work, we did get some permits to release natural enemies that were imported from native regions of SWD. Before releasing those, we did surveys in Georgia, and we did find some parasitoids of SWD, but these are 2 species that are more common, but they did not do much. We basically resorted back to releasing those exotic parasitoids that we have already released in Georgia, about 15,781 wasps at different locations in Georgia. And you can also help. We are actively looking for more sites to release those beneficial parasitoids. Our goal is to keep releasing until we get them established. And hopefully in the long term, they will help us a lot with control of SWD. chemical control, we do have lots of options to control SWD in terms of insecticides in conventional systems. Organic systems is where we are limited. Only spinosad is the best one, but we must rotate with other products to make sure we get through the field season in organic settings. These are just suggested control programs that we have used, but you can use any combinations of programs. Here is one strategy to develop combinations, is always use the heaviest, the strongest chemical first to knock down the population, and then you can follow with the medium to low efficacy products to get through the season to make sure you maintain good control. Also, insecticides, of course, do have non-target impacts. We want to make sure we try both. Even organic products have non-target impacts. So we did some studies and showed that reduced risk products may be expensive, but they are just as effective as broad spectrums and may have benefit for the long term in terms of saving those beneficials in the system. Also, when we are relying on chemicals, resistance can be a serious issue, and this slide is the best slide you want to keep in mind when you're making programs or making spray applications, to make sure use product from one class. Each class is color-coded here at the first spray, and then second spray use another class, a product from a different class, which will be a different color. And third time, if you need to go, make sure you use even a third class or a third color to make sure we don't develop resistance in these flies quickly. Of course, organic products are not as effective. What can we do? We have noticed that flies are active during dawn and dusk time, so making spray applications during those times is the best way to control, making basically the most out of our spray applications. Behavioral control. We have done lots of studies to develop behavioral control where we have developed products. This product, Combi Protect, is the most recent one that was registered last year. When you mix this with your spray application, you can actually get as good a control, even with a half rate of product, than you will get with the full rate once you mix this product with the spray. More work is still ongoing with this product. Cultural control is basically habitat manipulation. One that is the best way to control SWD is physical exclusion. I'll go quickly through these slides. We have shown these before, just to sort of remind you of these strategies. Physical exclusion, if you can, is expensive, good for smaller scale, but works perfectly. Irrigation, if you have a choice, drip irrigation is better because overhead irrigation creates more humidity in the canopy, which may increase SWD risk. Mulching, if you keep the covers on the ground, that helps any larvae that may drop out of the fruit to pupate in the soil, that kills those and overall lowers the population. Pruning, if you do heavy pruning of the bushes, it opens up the canopy, lets more light through the canopy to create an environment with a little bit higher temperature, a bit lower humidity, which makes environment less hospitable for the flies around the fruit. So if you can do good pruning. Harvest frequency also, leaving the ripe fruit out in the field means you're making the field more attractive for flies. So keep harvesting as frequently as possible to minimize the attraction of the field for flies that may be present out in the woods. And if you have a lot of ripe fruit on the ground, make sure you get rid of that by removing, burning, destroying. Either you can pack them in these clear plastic bags, or you can bury them in the ground deep to make sure the flies do not emerge out again. If you have harvested and you suspect that you may have infestation, storing at lower temperatures is also as a sort of a last resort to control flies, and also it gives you time to sell them. It slows the development of flies and potentially kill some of those larvae in the fruit while you store them at low temperatures. We have created lots of resources. Of course, this is Southeastern Regional Blueberry Guide, 24 guide will be posted soon. We do have My IPM app, we have Blueberry blog, and other extension publications from UGA. Here is one sub-page of the Blueberry Guide that you want to look at. We created this page this year, and the guide is, I think, page 52 of the Southeast Regional Blueberry Guide. You want to make sure you look at that. It sort of gives you a seasonal, at a glance, of everything you may come across and what you can do. To summarize, again, SWD is a key issue. I'll go through this quickly. And we do have some secondary problem. Sampling is the key when it comes to insect pest management. Stay on top of the system with sampling, and if you see any problems, you make sure you be proactive in making those applications to control them before they become serious issues. Here are a couple of reminders when it comes to making those oil applications. High temperatures can compromise their efficacy. Spraying immediately before, during, or following cold weather or freezing temperatures is also not good for oil applications. Do not use within 14 days of lime sulfur captan or some of those other products, because that does affect their, they may have some issues with phytotoxicity if you do that. Dormant oil applications exacerbate exobasidium, and current recommendations are to make oil application as early in the dormant period as possible to allow for as long as possible between oil applications and sulfur applications. With that, I would like to thank everybody, farmers, collaborators, and extension people. Quick announcement, sorry. We have a very nice webinar that is going to be on the February 7th at 2 p.m. Eastern Time. Here is a link to register for that. This will be a really nice update on organic management of SWD. This is going to be done by a national team of researchers that is working on organic management of SWD. And one last request, we are working on this national survey to determine impact of SWD and status of its management. Please take a picture of this QR code or follow this website. And also, I have a gentleman in the back. He has physical copies of the survey. We would really appreciate your help in getting this survey filled out. Thank you so much. And again, I'll be around. If you have any questions, feel free to stop me anytime. Thank you.
Thank you so much for your time and expertise, Dr. Sial. Welcome to Dr. Mark Sarnoda from UGA.
I'm the last one before lunch, too. I guess I'm the last one before lunch. Is that correct? I don't know. Okay, yeah, there we go. Hi, I'm Mark Zarnata. I do weed control work in small fruits, ornamentals, Christmas trees, and I like to tell everybody all the bastard crops that nobody else wants to work on. So anyway, I'm going to give you a quick update, a little different this year than going over spray guides and everything else, but there's been a lot of development or a few new things come forward that everyone needs to know about, and I'm going to talk about that, and hopefully you'll get some information in this 20 or 30 minutes. Well, I guess that's not going to work. There we go. All right, sorry the remote's not working. We'll try to make it work here. All right, I'd like to ask, how many people here have used Surfland before for weed control? Okay, well, that's how many of you have been able to get it here in the past 2-3 years? Okay, well, it's gone, and I don't think it's going to come back. There was some hope there when it first, I don't know if you know, the plant burned down, the last plant that was making the active, and I don't think it's going to come back. So we'll just say it's pretty much, if you find any, just use it up and be happy you had it. It was a great product. It was really safe. And the big thing about this product, it wasn't the best weed control product, but it brought safety to the table. So I've been telling you for the last 2 years that we were going to get dual magnum. We finally got dual magnum. And so the rates that they gave me were 16 to 21 ounces per acre. It provides really good weed control of small seeded broadleafs, and it's a 24C label, which I'll talk about here in a second. But if you're going to use this product, you have to go to their website. And that's the website right here. And you have to go and fill this. It takes about 5-10 minutes to fill that out and basically waive your rights that if the product causes damage, that they're not responsible. It's a Group 15 herbicide, needs a tax mix partner really. So if you're going to use this product, you need to mix it with either Surfland or Trellis. And we got it in only blueberry and blackberry. I tried to get it in peaches, pecans, and a few other crops, but they're not budging on that, I don't think. So when you go to the website and fill out that form, it'll actually give you the label. It's going to look just like this. This is the first page, and all the information there that you want will be on this page. So, and we have it till December 2017, and if I'm still here in 2017 or 2027, I'll reapply for it, and it's much easier to do a reapplication than it is to submit an original one. I can tell you that. It's very difficult to get credit for these type things for when I do this type work as an extension specialist. Kind of upsets me. But anyway, we spent two and a half years to get these with a lot of phone calls, a lot of research goes out, and I wish the administration would understand that this is a very important thing that we do, and when we need to get credit for these type things that really help the growers out. Okay, we did get the herbicide Break On last year. It's a great pre-emergent herbicide, another good one for rotation management. It's a very different mode of action. It's fluoridone, if you're familiar with sonar and weed control in, what do I want to say, in aquatic situations. It works really well in that situation. And anyway, sorry, the microphone's so short and I'm a little bit tall. It's really good in controlling many broadleaf weeds. Pigweed was the main target that they released this product, and it was mainly used in cotton. It's called Break in cotton, Break On in small fruits. Its PHI is 30 days, REI is 48. It's a Group 12, and in 2023 trials that I did, I saw no damage in any of the mature plants that I worked with. It's the prettiest herbicide I ever worked with. You can see that pretty turquoise color. Did not realize it would be like that, but an excellent pre-emergent herbicide. It smells good, and it actually is a pretty herbicide. Like I said, it's the prettiest one I ever worked with. Beautiful turquoise. So as far as the rotation is concerned, or how you'd use it, instead of Surfland, you're going to use dual magnum simazine and then maybe dual magnum gallery, and your first application and maybe your second would be with Chateau if you got bigger plant material. With Chateau you want to have plants at least 2 years old or older. I like to say at least 18 inches. And then the second year you could do dual and simazine again, Chateau, and then you could kick in the Chateau and maybe Solacam. But this product is really only going to be good for really young plant material. Once the plants get a little bit bigger, you need to switch over to better, more active herbicides such as Chateau, Solacam, Allion, Carmex, lots of choices. As far as the cost is concerned, Dual's going to run you, it's about $50 just to buy the product sorry. Oh yeah, that'd be much better. Thank you. Does this need to be turned off? Okay, just turn it. We have an off switch. Oh, thank you. That's much better. I can't tell you how many times I have to do this when I always get these little microphones and I have to bend over and then I'm constantly getting feedback and everything else. But anyway, as the cost you can see is about $50 for a 20-ounce rate would be what you'd have to pay for it. So it's very comparable to Surfland, and it's going to be an excellent fit for us. So if that's what you're looking for, this product's going to fit. We have a 24C now in Georgia, and it's both in blueberry and blackberry. Okay, now I'm going to talk about a couple herbicides that people still aren't using enough of. I still run into people using that are unaware of these and just don't know about them. But Zeus or Zeus Prime, an excellent product. If you have yellow nutsedge issues, this is a really good choice to go to. Highly recommend it. Highly recommend this product for pre-emergent weed control. It's a really good choice, replacement for Chateau. If you have yellow nutsedge, this will give you some pre-control of that product and early post, and I can't tell you to really push people to use this product. It's Zeus is just straight sulfentrazone. Zeus Prime has a kick of cofentrazone in it. I really don't see any additional, in the test that I've done at least, I haven't seen any additional benefits from that, but sometimes you can't get Zeus by itself. You'll get Zeus Prime from your distributor, and that's fine to use that product. It's a, let me see, Zeus, the max rate you use would be at 12 ounces at 6 to 12. Recommend you use at least 10. Zeus Prime, I tell you to use at least 12 ounces on that product, at least, and you can see the rate here, 7.2 to 15. It's a PPO inhibitor. Let me see. And like I say, I can't stress the importance of yellow nutsedge help, because we really need help with that weed, and I'm sure you're all familiar with yellow nutsedge. Not really good on purple, so you have to learn to know the differences, but it works quite well. Remember that's not working? Okay. Allion. A lot of people still might be a little hesitant to use this product. It's really good. Only use this on plants that are 2 to 3 years old at least. And the other thing is, I'd tell you, do not use this product over and over and over again. I sat here and watched talks about people mixing modes of action. This product we have no long-term experience with. There could be some root inhibiting issues with this product. It moves into groundwater a little bit, or into soil a little bit, so it can cause root damage. So I'd highly recommend you rotate this product with some other materials, and use this. I'd like to see this product used in the fall also to give you the longest amount of weed control. So if you're looking for a window where you need the longest weed control, this is the product you want to be using. Let's see, Twin Star is another one that, let me get my phone here. Keep track of time. Sorry, Ash shaved off 12 minutes of my time. 10 minutes of my time. I have to talk to him about that. But anyway, I'll try to keep track so I don't go over here. Quinstar, another one people don't know about. This is a really good replacement for Stinger or other products you might want to use for controlling broadleaf weeds. This one will get dog fennel and several other broadleaf weeds. Excellent post product. It's a really strange herbicide. It'll kill some grasses post-emergently. Works good on, for example, torpedo grass, if you're familiar with that. Real difficult to control. It's a group 4, which is growth regulator, is where they classify it, but it's got some other modes of action in it also. But the only thing is pour on the yellow nutsedge. Embed Extra, this is the 4D choline formulation. It's the low volatile ester formulation. Another really good one. People are a little scared to use this. I try to tell people back when before I was born, this 2,4-D was labeled on everything. And as people got more litigious and more worried about damage, kind of shied away from this product. It's out there. Encourage you to use it. Use it on some small acres if you're scared about it. Controls broadleaf weeds really well in blueberries, and blueberries are fairly tolerant of it. It's a 3-pint max. Let me say embed. It's used at 3 pints or quart. I'd like to see it used at if you're going to spray. Got to have a surfactant with this also. And of course, it's a growth regulator product. No control in yellow nutsedge. I forgot to get rid of the labeling there, but Hulk is a new one. What we're going to get next year. Sorry about the embed. I could take that out on how that came over. But this one is one we're hoping to get next year, the year after. This is going to be a really good replacement, and it's going to control some really unique weeds. This also is a growth regulator type herbicide, and it's poor on the yellow nutsedge, but it's going to give us a really good uses for excellent product for controlling broadleaf weeds selectively, which is going to be a really good use. I got a hold of this product back in September, put a couple quick trials out. Was working on another project, noticed this guy had some tropical spiderwort. I don't know if you have this problem or not, real difficult weed to control. I tried this product at a pint product per acre, and you can see it's worked really well. Controlling tropical spiderwort, which I have a picture up there in the left right-hand corner. I sprayed in between the rows too to try to help the guy out. But you can see the tropical spiderwort can kind of vine itself up into the blueberry plant, and this was doing a really good job controlling it. I didn't see much damage on the plant, but of course I'd like to go back and look at these plants when they flush out this spring, see what they look like, because it usually takes about a year to get information on that. Another issue I constantly am saying is people are constantly using glyphosate. This particular grower, I've run into several of these this year, had issues where people they'll put glyphosate out on every spray. And if you do that, you can get away with a spray of glyphosate on larger blueberry plant material, but at some point you're going to start manifesting damage. So basically what it is, you know, think of glyphosate as a poison. It's a herbicide. The plant can take a little bit of that, and if you get over it, probably in the plant over probably 100 parts per million or something like that, if I start doing sampling on the plant material and you start getting above that average, you're going to start manifesting damage, which means you're going to start seeing symptoms like this. You can see where the buds are starting to get deformed, not coming out right. This is real characteristic of Roundup damage. You start getting the rate too high, the buds aren't going to develop. You might think it's a root rot or a disease. This is where people need to start asking questions. And this is where we all kind of work together. Jonathan and I have to talk a lot sometimes because he'll get damage and it'll look like some fungal damage, but if he starts asking questions, it ends up being some herbicide damage and it comes off to me. So I could show you a whole seminar on damage that I've seen. But this damage also looks very similar to things like triclopyr and some other things, unfortunately. But if you start asking questions, this grower is using glyphosate. Now, he's right on the tipping edge on this field where he needs to not use glyphosate for 2 or 3 years, is what I told this fella, and hopefully the plants will start growing out of this. But if he's continued to go on with this, the plants are going to start dying, really poor production, and all that. Everything's going to go downhill on him. But you cannot use glyphosate on every post-emergent spray. A lot of guys think you can use it, and it's fine. I would not do that. If you're going to do something like that, you want to use something like glufosinate or Rely. There's a whole bunch of names for it, Cheetah, Reckon. It's a great cleanup herbicide, but wouldn't recommend using it more than once a year or every other year when you have to come back and use it like a rescue treatment, is what I try to tell people. Glyphosate's flow and loaded. It's completely systemic, so you have to understand that this is a systemic herbicide in plants, and it can be root absorbed. So roots can absorb glyphosate, and this is a key thing. If you keep spraying glyphosate on sandy soils here, glyphosate bonds irreversibly to the soil. But at some point, it's going to start running out of docking places on that soil. And once that happens, it's going to start getting loose in the water profile, too, which is the free water matrix, and that's when you can start having problems. So anyway, I always try to tell people, if I take glyphosate and I water a tomato in sand and continually water it with a spiked solution of glyphosate, you're eventually going to show damage, okay? Dicamba. Hopefully you all listen to this. So I'm very concerned about this product. I hope it never causes a problem in blueberries, but the more I'm in South Georgia, the more I get a little bit concerned, and hopefully you can listen to me, and hopefully if you have any problems, you need to identify this quickly, and hopefully it won't prevent any issues from happening. We have cotton, soybeans got genetically modified. The products they can use are Extendamac and Enlist. Cotton growers can use up to 2 quarts an acre, 2 pounds active per acre. Dicamba has a KOC of 2, which herbicides have KOCs from 0 to 1,000. Anything below 30 is going to move. That's all I can tell you. So, and it's the interaction, how it interacts with the soil. pH depends on this, and there's several other things, but dicamba is going to leach. The half-life of dicamba can be anywhere from 3 weeks to 9 months. It gets loose in the water, it goes up. That's when you start getting 9-month periods. So if it doesn't have any chance to interact with soils, it's going to last a lot longer. And 100% of the soybeans and cotton we can now be a full 100% GMO crops in our state. So I just worry, and the thing that bothers me, it's not deep well, it's collection water. The reason I say this, I do ornamental work. That tree right there is damaged by dicamba. And I can't tell you the hundreds of trees I've seen like this where people spray the turf grass around that tree, and in a year or two, this starts popping up. So, and that tree's not going to come back. They're going to have to rip that tree out. This particular incident was in a closed community, and this guy sprayed a couple million. He probably had about $3-4 million in damage that he caused because they were just spraying basically Trimec. Every 3 months he was going out there spraying it, even though there was no weeds in the turf grass whatsoever. So anyway, he learned a lesson from that. So anyway, blueberries, this is one where I was just doing stump applications. One application I did trying to control oaks and pines, and you can see the damage that caused on the blueberries, and that's just from the dicamba leaching out from the cut pines and oaks and being reabsorbed by the blueberries. You can see how detrimental that can be to the plant, and that's not a direct application to the plant itself. You'll see twisting like this. It's real common. And then this picture here is, this could be ground zero, but like I say, I hope this doesn't happen. This is all I'm just trying to avoid this. What it is this is almond. There's 2 lakes here, and every white patch here, it could potentially be cotton. So, and then right in here, we have blueberries. So we have all these places. Here's some cotton. Right here's a lake. This is irrigation lake right in here. So, and a lot of times there's even drain tile coming out of these cotton fields right into these lakes. So as a weed scientist, and if that's a 300-acre field, you're going to tell me you can spray 300 quarts of dicamba, and you keep doing that, here and here and here and here and here. Is that going to get in that water or not? Well, we don't know, and the problem is no one's testing for it. So that's what bothers me. And again, it's just in these type of locations. So one of the problems, I started collecting water samples. I got some funding off the Blueberry Commission, and I collected some this past year that I was certain I'd find some dicamba amounts in. And so they came back negative. And so what I decided to do was spike one of the samples I sent in to see how good the lab was that everyone's using. So I spiked it at 100 times the fluoride detection, and unfortunately it was missed by that collection lab. It came back negative. So you can tell you right now that doesn't make me feel good. And there's all kind of reasons I could talk about probably why that happened. So I'm trying to work it out with the lab a little bit, but they didn't want to discuss it too much, but that's a problem in itself. But what I think happened is it bound to the soil particles. So when they filter that product out to run it through HPLC mass specs, it misses it. And then, that filtered water, that unfiltered water is still active. So if I take that unfiltered water and water tomatoes or for some indicator plant, blueberries over time, they're going to flash damage. So next year, I'm hoping to collect 2 samples a year, 2 times a year in August and September at key locations, collect water and pond sediment samples, send water samples to lab for testing. And I want to do some bioassay experiments because I'll show you a picture where plants are just as powerful as HPLCs and GCs to detect this. And I'm going to use cold crops, and tomatoes can detect things down to 10 parts per billion, which is as good as any of the HPLC or mass specs. And I can do it a lot cheaper. Instead of $200 a sample, I'll do it for about $5 a sample. So I did a little work with dicamba. I watered some tomatoes with 100 mils 3 times a week with either 10 parts per billion or 100 parts per billion. And in 3 weeks, I was flashing damage in tomatoes. So it doesn't take very long. This would be an example where I use a cold crop like cabbage, or this was kale. And basically, you either use, this was a contaminated soil. And I planted that. And as you can see, within 2-3 weeks, the kale coming up and showing damage. And this is how I tell people if they ask me if they sprayed a herbicide and they want to know if there's any herbicide damage or that could damage your blueberries. This is something you can do in your own greenhouses. You can just gather that top 2 inches of soil, put it in these just a pot, grow it in the greenhouse in a clean soil, and compare them. And once you get the 2 plants that look the same in the same soils, then you're good to go. This fella here, he ain't good to go. So, I can tell you that. Anyway, but that's it. Sorry, I'm out of time. And if you have any questions, I'll try to entertain them. There's nobody after me, right? There's nobody after me, correct?
No.
Okay.
There's nobody after
Yeah, go ahead.
Casseron and 45-year-old rabbit eye plant. Perennial weeds are really a big problem.
Yeah, he's asking about, everybody heard that, he's one about perennial weed control, and Casseron is a herbicide called diclobinol, which we have. Unfortunately, that herbicide, it's pretty poor herbicide as far as control. It has some really unique things to control particular weeds. For example, you can control Florida betony, and if we can use the sprays on it, you can control the Japanese climbing fern. But for perennial weed control, Quinclorac product I mentioned, the Quinstar, would be a real good choice for you to use that. Yeah, excellent. And you can use glyphosate too. Don't dismount glyphosate, but you can only use it, pull that product out of your tool bag only when you need it. So if you had a situation where you had a blueberry farm, for example, that was not being maintained for a few years, you could go through and cut out the weeds, and then you start getting a reflush. You can use glyphosate to paint it on the oaks and pines to get rid of that if you're trying to rejuvenate a farm, or you can use it as a direct spray to spray some of the weeds out on, as long as the blueberry plants are real barky and corked up. You can do one application with that with even a pint to a quart, and you get away with that one time. That's how you clean it up. But the Quinstar I mentioned, that's a really good product for controlling vegetative perennial type weeds that you're trying to get rid of. It's fairly safe on. You can get really sloppy with that product, and fairly safe on the blueberry plants. Really good weed control. Did you hear all that? And you can and anything else? Yes, sir, in the back. For chemical mowing, for glyphosate, you're talking about row middle? Yeah, row middle. Yeah, I usually have slides about that, but that's a real common practice. If everybody knows, a lot of times people use Roundup for chemical mowing. There's some problems with it, but it also works real well. If you have Bermuda and Bahia, basically you spray glyphosate at about a quart per acre, and it'll get the Bermuda and Bahia, in between the rows. Really, you try to keep the plant sick is what you're trying to do. You don't want to kill it, but just keep it sick so you have some cover. The only problem with that is if you start getting weeds like Roundup-resistant pigweed and some other weeds that are tolerant to it, all those weeds are going to start growing. Johnson grass, so they don't play by the rules, and you're going to have to go in and spot remove those with other products. So you'd have to use a pre-emergent herbicide, for example, for the pigweed, and then for the Johnson grass, you're going to have to use something like Certainty or something to control that post-directed. Oh yeah, it should be fine. If you're just doing it in a row middle with a shielded sprayer 2-3 times a year, I think you'd be okay. I don't particularly like it but usually, usually those are in real sandy locations. So, but you do have the potential where the roots could creep out into from the blueberries on the hill into that area you're treating. Or you got to remember, the glyphosate can capillary up into that median too, or into the alley where the blueberries are growing in the row. So there is that. But if you're using usually glyphosate applications for that, you're going to use 1 to 3 applications a year. So you put a quart out, and then you come back with a pint. Maybe once you start seeing regrow, you can put a pint out, and then it usually beats it back enough, and that's a low enough rate where it'll help keep that under control. But that's a good question. Yes, sir. Not that there is some pre-emergent activity. He asked about Quinstar, whether there is a residual with it. There is a residual with Quinstar. It'll last you'll probably get about 6-8 weeks with it. So they use it as a pre-emergent. It's a really strange product. They call it Drive and turf grass. It controls, and the big thing in turf, it controls torpedo grass post-emergently, which is really difficult to control if you ever have to deal with that. But it will control crabgrass, I think southern, or large crab, not southern so well, post-emergently, but it also provides pre-emergent weed control for, I'd say, at least 6 weeks or so you get out of it. So it's a really good product, highly underused. And again, I always tell people when you have these new products that I suggest or I've worked with, try it on a small acreage first before you go full bore and spraying it on 100 acres. Try it on 10 and see what you think. Because, I'm not here trying to sell you the product. I'm trying to give you another tool to put in your tool bag to control weeds. Anything else? Yes, sir. For Bermuda grass? For killing it or knocking it back like the fellow behind you? You're trying to control it? Yeah, probably Select Max is the best post-emergent grass herbicide, that or Fusilade. Post or Post Plus, they need to reformulate that product in my eyes. The surfactant load is not good enough. It takes too long to see activity. I'll spray Post Plus out and it'll take a week before I see the plant start dying. If I spray Select Max out, 3 days, it's dead or dying very quickly. But they're both good. Yes, sir? You need to do active growth. So preferably in the fall is always the best, man. If you get a fall application on that, really, when that plant starts taking in stuff to make it over the winter, that's a really good time. Glyphosate's the best product, the cheapest products you can use as long as you get the rates up about 2 quarts. But you don't want to do that around blueberry plants. So that's where Select Max, you really need to use that product. If you got it growing up into the blueberry plant, that's a great product for it. Yes, sir. Select Max should be fine. A lot of people might heard, I guess you're talking about Arrow, where it was causing bud blasting on the leaves. That was because people were spraying vasi grass that was 6 feet up in the plant using a air blast sprayer to spray it into the canopy of the blueberry plant when the blueberries are setting bud. And then they wondered why they had some bud blasting on that. And it really didn't, I don't know, there's some people in here like Gerard you could talk to, but I don't think that affected yield that much. But you could see, I had pictures where you could see the guy was using Select Max and switched over to Arrow, which had a different surfactant load, which heated up that product, and then for some reason was causing damage to that developing bud. Because what those herbicides do is they jack with the fatty acids when the cells are getting put together in those buds, and that was messing those up, which normally doesn't happen in broadleaf weeds, but if you get the rates up and put it on at the right time, at the right rates, it'll work. Which one? Which one now? Dual Magnum, we got a label for. That's us. Yes, sir. Florida, last time I heard, no. I talked to the weed scientist down there, Gerard. Peter, thank you. Peter, he won't call me back. I don't know. I can't get him to respond, but he should submit that. And the problem is they won't let us use data from other states. They used to let us do that. So now the extension specialist, whether it's 24C for insecticides, fungicides, or herbicides, that you have to generate that data in the state. And you need 2 years of data, safety data, to go forward to say it's controlling this with safety, and then you'll get to the Section 24C. And then you have to deal with the Department of Ag that's so woefully understaffed, you can hardly get an email back, and it's a fight with that. So you got the EPA to fight, plus you got the Department of Ag, and then once they send it out, then it's good for, what's it, 4 years, I think, and it's got to be, but when I reapply for it, it's nothing. I just basically write a letter saying we still need it. Yes, sir, Gerard?
How much Carmex can we use in the aisles where we want clean culture before we get into a problem? We use it like twice a year.
Yeah, I wouldn't see a problem with that. Yeah, I wouldn't see a problem with Carmex. I mean, it's been around for so long. I know you can get some problems on blueberries when you're spraying it underneath the plants, but twice a year in the alley, I wouldn't think that'd be a problem, particularly with the shielded sprayer. And there's not enough root pickup there. I don't think you'd have a problem with that. It's a PS2 inhibitor, though. It can be root absorbed and all that, so you have to be careful. But you monitor your plants. If you start seeing your plants flagging a little bit or yellowing, you need to lay off of it. Is what you got to do. But that's an excellent choice for. But you can always rotate chemistries too. But of course, it's the cheapness of it, because Allian and Chateau are a much better choice. But of course, the price is a lot higher. Great question. Anybody else? That it? You can ask me questions. I'll be around and be happy to try to answer them. Thank you.